Okay, thanks, Susan. So I was asked to give this talk by Carrie Ann and Sharon, who were unable to be with us this week. So I'm stepping in and pinch hitting. This is not my area of expertise necessarily, and I am not an expert geneticist. There's people in here who've probably forgotten more about genetics than I'll ever know. So if I say something wrong, I invite people to stand up and correct me. I'm going to try to get through what I've got and was given to present, and we'll see how it goes. So with the Great Lakes program, we're looking for AIS detection. We're involved in an early detection and monitoring program. And with this, we've got 4 different offices throughout the Great Lakes working on this. And those are fishery or FWICO offices working on those. We've got the Ashland office, we've got the Green Bay office, Alpena, and the Detroit substation, part of Alpena, and then the Lower Great Lakes office. So among those, we're doing organized sampling across all 5 Great Lakes. And then you see my little microscope things here is where our labs are located. We've got the Whitney lab in Onalaska, Wisconsin, and the Lamar lab in Lamar, Pennsylvania, where our samples go to get processed and analyzed. We're trying to do is early detection before the species that are invasive get established and start spreading. So as long as they're localized and rare, we actually have a chance of doing something about them. We don't want to get to the point where they start spreading throughout the lake and are out of control, and we no longer have an opportunity to do anything about their spread. Because we would like to be able to do some kind of eradication and management effort, but we can only do that when they're at the front end and at that kind of small population size. So once they start exploding, we're in trouble and we can't keep up with them. To determine where to go sampling, they've set up a bunch of what are called high priority locations, and these locations are based on a number of factors. We've got shipping traffic, we've got connectivity, meaning potential waterways, rivers, especially Chicago with the Chicago area water system. Population centers, large urban areas tend to have a higher potential for introductions. Recreational boating, meaning potential for accidental boat introductions, transporting between different lakes. And a fancy word, propagule pressure. And I thought this was actually brought up in a good example yesterday when it was spoken about pink salmon and having a large release through the water sewer outlet into the lake unintentionally. And that's a large propagule pressure event where a large number of invasive or non-desirable species are released or accidentally introduced into an area. You could also have two. You could have one male and one female, and that would work as well too. The amount of pressure, though, determines the potential for spread and the rate of increase over time. I'm just going to focus on Lake Michigan today, but I did mention this is happening in all 5 Great Lakes, but I'm going to use just data from Lake Michigan. That's what our office works on and what I was given by Sharon and Carrie Ann to present today. So in Lake Michigan, we've got 9 of these high priority locations, and throughout the Great Lakes, we're currently focusing on the top 25. For reasons of logistics and resources, we can't do all of them. So 25 is our target right now. If we can do the top 25 across all 5 Great Lakes, we think that's our best use of resources at this time. And Lake Michigan has 9 of the HPLs, high priority locations. Now I only have 7 on here, but number 1 down here in Chicago actually has a combination. There's 2 more in Evanston and Gary that aren't labeled on here, but they count as well. So there's 9 of these locations in Lake Michigan that rank in the top 25 that we do sampling at. Originally, we started working on just traditional sampling and trying to find ways to, oops, what's going on? Okay. Doing traditional sampling, finding ways to collect them in the normal manner, doing electrofishing, netting, trap netting, gill netting, things of that sort. And that's the standard way you would collect fish for invasive species. But with the advent of eDNA, we're able to do a whole lot more sampling of species than we ever used to be able to, cover more water, less time, even less resources. And it's really been a great addition to the program. Now, prior to the last couple of years, have been focused on quantitative PCR. This is direct species sampling. We're looking for particular individuals, or not individuals, particular species that are of interest. Over time, though, we've developed a new metabarcoding project that we want to do, and that is looking at a community. So it's a different way of approaching it. I'm going to get in a little bit of an explanation here of how those two differ and what they're able to both provide. It's only the last year we've been able to start doing the metabarcoding, and it's been a great addition to our program. So first of all, most of you probably know what eDNA is, but for those of you who don't, what is eDNA? We're looking for genetic material that is shed into the environment. And this could be from scale, feces, mucus, urine, gametes, skin cells, carcasses, basically any type of organismal material that is left in the environment. And it's actually quite shocking, the small amount of material that can be picked up and collected and then used for analysis in the lab to detect the presence of a species. This can be transported by air, by water, by sediment. When I say water, obviously that's being transported through direct contact to the organism that lives there. Sediment, things get built into the sediment, they get stirred up, and then they get released. We can pick it up that way. And then in the air, we're going to use this as like birds. Believe it or not, birds are a way of moving this around. And I was surprised with the amount of avian traffic that actually contributes to the distribution of eDNA. Also, it's interesting that eDNA is now considered a gear. Typically, as fish biologists, we think of a gear being something you carry with you and you throw out and you deploy, and you're collecting an actual fish and have it in hand. With eDNA, you're not actually collecting any fish themselves. You're collecting evidence of the presence of the fish. But we still call it a gear as we've added it to our collection protocols. We use it because sometimes traditional gears may be ineffective. We have cryptic and invasive species that may need to be used, or where eDNA can be used to compensate for the inadequacy of traditional gears. When we have low density, a rare species, eDNA can be very effective. And various life stages. When we have larval fish, this comes in particularly helpful. They're difficult to pick up. We're not going to see them frequently, but larval fish can be present, and evidence of them through eDNA can be used to detect them as well. Also very difficult habitats to get into sometimes. You can't get a boat in somewhere, but you can get in and get a water sample. And then also difficult to identify species. Again, I'll use the pink salmon thing from yesterday. Perhaps we don't know a Chinook from a pink at a certain life stage. Here it doesn't matter. We've got DNA. There's no doubt which one is which. Also, when you're looking through a whole bunch of species, myriad of minnows that exist out there, shiners, different things that look very similar at small sizes, get detected very precisely using eDNA. So how do we get the eDNA? Currently in our program, we're using two methods. With our qPCR eDNA, which is part of the program, we're just doing a dip and centrifuge. We dip these tubes in the water, we collect water samples. I did this last year, so I'm slightly knowledgeable on this. The amount of sterilization to prevent contamination from this is incredible. The number of gloves we go through is fascinating. So that, and then bleach. We order bleach by the 55-gallon barrel to keep things sterile out on the boat as well. So I'm not kidding. It's really that much bleach we're using. So a lot of decontamination to be able to prevent this. And every day when they get back from the field, the boats are hosed down and prevented from having any contamination as well. So the potential for contamination is high, and we do a lot of work to prevent that. Now, metabarcoding is what I'm going to be talking about mostly today and focusing on and explaining why it's a great tool to have. And for that, we actually use a Smith Root designed backpack sampling unit. It's got filters, and the filter is the portion on the right here. It's a very small thing that gets added onto the hose. You suck the water through there. It's all programmed for the volume, the amount needed. And it takes anywhere from about, anywhere from 5 to 15 minutes, depending on what you're looking for and the size of the sample that you want to get a sample into the filter. Once you're done, you pull the filter off, stick it in a sealed, sterile bag, and put it in a cooler and send it to the lab for analysis. So at the lab, I saw those tubes before. The tubes come in by the thousands and get sent to the lab. This is our Whitney Genetics Lab in Onalaska, Wisconsin. They get them and they do the analysis. So the one on the left is for the qPCR, and the one on the right, I'm going to plead ignorance. I don't know whether that's doing qPCR or metabarcoding, but it's doing genetics, and I'll leave it at that for now. That covers it all. And again, I'm going to keep this very high level, or well, high level is the right word, very 30,000 foot level. I'm not going to get into the details of how the genetics part of this works because I don't know. And I'll try to just talk about the application and why it's important. And if we have people, I saw name tags for both Pat DeHaan and Meredith Barton being here. So hopefully they can maybe talk about that later on if I run into them. They're the experts on this. Anyway, we get the samples here. Our folks in the labs are fantastic. They process thousands of samples. So it's not just the Green Bay office. This is all four of those Great Lakes offices are turning in thousands of tubes and filters to get analyzed. And we get our data processed through there. And from there, we're able to reveal what species are out there. Now, I mentioned that qPCR, that's looking for species that are specific. We actually have a marker specific for silver carp and for bighead carp, and a joint marker used for bighead and silver carp. But for the metabarcoding, we're looking for a large number of species. I'm going to mention how that works here. First, though, when we get these results back from the lab, what does it actually mean? There's maybe some confusion sometimes that when we get a sample, you say, aha, we got a positive. That means there definitely is a silver carp or could be anything, a northern pike, right in this area where the sample came back positive. Not necessarily. What it really only means is that that DNA was found there at that time. And how it got there could be through a number of different processes. So the common questions are, when did it get there? Answer is, we don't really know. All we can know is that the rate of degradation of the eDNA is anywhere from sometimes hours up to days, maybe at the outward or the outer edge, maybe a week. So from that, we are able to kind of extrapolate and figure out within a week or less, probably that fish was present in that area. Were they live or dead? We really don't know. Perhaps the amount found or evidence of a carcass could help with that. Were they stationary or mobile? Meaning, was this just a very sedentary fish, or was it something passing through or migrating? Again, we really don't know. All we know is that it left DNA there at some point. And what does it say about abundance? Again, really not a whole lot. If there's a whole bunch of eDNA, most likely it means that you've got a school or several individuals there. If you've got a very sparse amount, then most likely you've got a few number. But where that line is, it's not a linear relationship, and making an inference is extremely difficult. How does it move around? I kind of mentioned before, it could be by boats, could be birds, water current, predators. We could have something eat a fish, poop it out, and then the eDNA comes out and you can detect that. So there's a potential that something was eaten, carried around by a predator, and deposited somewhere else. And even wind will push it around quite a bit, we found. And in Chicago, you can imagine, we get a lot of wind down there. We even had one instance where we detected carp DNA after being in the gut of a bird for 8 days. So it can last for a little while. Target organisms, the actual beginning of the program, we were focused on bighead and silver carp. That was our primary target for this we were looking for. But we've since added in this metabarcoding, which is what I think is the nice new addition to this. Now we're looking for multiple species, and this really helps out with trying to figure out what kind of communities we have out there, just in a general setting, but then also especially for invasive species. There's invasive species we may not even be aware of have been introduced, and this gives us a way to kind of detect anything that's out there. The PCR eDNA can be used, like I said, for targeted species. It's loosely related to abundance. Like I said, we don't know how much, but more eDNA typically means more fish, or at least more time spent with a fish. And then the metabarcoding, we could do more with biodiversity, ecosystem health, diet. Actually, diet is a fascinating part of this. I was even, in my former lamprey days, involved in a thing where looking at the stomachs of sea lamprey that were captured in traps, taking out what was left in their stomach, and then analyzing the content of adult sea lamprey caught in traps to figure out what they were last feeding on as their last blood meal before they went into the traps. So you can find out what's in the stomach of a fish, run a eDNA metabarcoding panel on it, and say, oh, it looks like they were feeding on X, Y, or Z. And then novel and invasive species, again, what the real focus of our program is looking for. Advantages, the PCR, we're looking for specific taxa. It's much more sensitive. We can pick things up. We get results quicker, and we can look for, it's also non-invasive. We're not needing to collect a fish and take any kind of tissue sample. For metabarcoding, we're looking at communities. We're looking at broad taxonomic groups. It's much more cost effective versus the qPCR, and it takes a lot less time to get your bang for your buck, the amount of effort put in and the data you get back. And it's non-invasive. We're not having to collect individuals and take any tissue samples. Limitations, the qPCR is more costly. We need specific targets, or target markers that are developed by the lab and the geneticists. And there's a potential for false negatives, meaning that you detect something that's not there. Which has a false positive. They're not there, but what am I trying to say? They weren't detected when they actually were there because the signal wasn't strong enough to detect it. eDNA metabarcoding, we need large amount of database, and this is always increasing. I talked to one of our folks at the lab recently, and they said, yeah, we're continuing to work on adding this species and that species. They're looking for fish biologists around the country to send in voucher specimens that they can use to continue to build the databases. And the library of fish that are available to compare against keeps on growing every year. However, they're more difficult to detect at low abundance. That's where qPCR is better. Metabarcoding is better when you've got a large amount of eDNA present in the environment. And like qPCR, it's also subject to false negatives. And we pretty much use it for presence and absence only. We're not going to get any information on density with metabarcoding. And it's not as sensitive as it either. So with qPCR, we started off like this in 2013. We looked for silver and bighead carp only. 34,000 samples were taken in just Lake Michigan alone. And we have all kinds of ways of going through this and getting the information out of it. I wanted to show here's where this is taken for locations around the Great Lakes. And then in this little box down here, we've got the Chicago area water system, a few positive samples just using qPCR. This took a lot of work just to find these few samples here. In comparison, we've got eDNA metabarcoding, and over time, we've been able to use information, learn about this, apply it to what we're working on, and find ways to develop a new approach in the Great Lakes Basin to looking at more species. We started this in 23. Here's just an example in Milwaukee, the Milwaukee River coming in here. And we've already been finding positives for grass carp. These blue dots are just positive grass carp samples using metabarcoding only, whereas that would have taken a lot more work, especially with traditional gear. We're always looking to improve our efficiency and effectiveness. And this here, to me, is kind of the take-home slide. I think this is the most important thing to look at. If you see the amount of effort put into this with traditional gear, 816 units of effort across 12 years versus 175 units of effort in 1 year. And we got more information from metabarcoding in that 1 year than 12 years of traditional gear. And we got more unique species from it as well. So it's a powerful new tool. And I see I'm running out of time. I'm going to skip through really quick here. These are just the species that we're finding out there. Don't need to look at all these, but just give you a size. Wanted to talk a little bit about things that affect this. A lot of things affect the variability, which makes it difficult to get through eDNA. Environmental factors create a lot of variability in the samples. Where you're sampling it makes a lot of effect, whether you're in a lake, whether you're in a stream, a pond, a big river, a small river. And then something our office is working on is even microhabitat selection. Where do you sample? How do you maximize the potential of collecting something in the environment? And both the lateral fate of the eDNA, meaning side to side, it's foul wag on the bank, and then also the longitudinal fate, headwaters, tailwaters, whereabouts in the stream, doesn't matter, makes a big difference. From there, we're going to hopefully get some information and data that will help us determine where to take the best samples and increase this even more. And then for a summary, just want to say that metabarcoding is a great new addition to the EDM program. We were only using traditional gears and qPCR before. Now introducing this opens up a whole new wide array of possibilities. Early results show that we're able to find a lot more organisms with a lot less effort. It's important to recognize it's not a replacement for qPCR or any other gear, but it's a nice complement to go to it. All these factors, like I said, affect this, and we have to account for these and deal with them in some way on different rivers and different areas. Sampling Chicago is not like sampling Milwaukee, so there's a lot of differences. Like I said, it complements it. It doesn't replace anything. And then lastly, we're still looking at ways after the first year of doing this to fine tune it and try to determine how we can better use metabarcoding in a way to effectively sample the environment and get even more information on invasive species and the whole fish community in general. So with that, I'm going to put up the contact information for Carrie Ann and Sharon. Contact them, not me, for more information. They're the experts on this. And I'll try to do any questions if you have them.